Hi everyone. This is the practical approach part 4 to the identification of commonly encountered organisms in the clinical laboratory, focusing on the gram-negative bacilli. All right, so let's proceed with the identification of common isolates, now focusing on the gram-negative bacteria. And once again, characterize the shape of the organism, whether it be a cocci or rod, or we call them bacilli. When it comes to gram-negative bacteria, we'll have to admit that it becomes more challenging to describe its microscopic morphology, except for the Neisseria or Moraxella species. We have already talked about this, but as we will cover later on, you may find that certain gram-negative rods will appear and could be mistaken as gram-negative cocci. So first and foremost, you have to determine if they are rods or cocci, as I mentioned before. Some gram-negative rods are quite easy to tell. See image 1 and compare with image 2. Both are gram-negative rods. Now compare images 3 and 4 with 1 and 2. See the differences? Nevertheless, when you get to learn more about each of the different species, there are certain unique characteristics, even in their microscopic morphology, that you would eventually become familiar with. And this takes practice, practice, practice. Looking at the images, looking under the microscope when you get to the clinical laboratory, and be able to correlate a particular organism with a specific morphology. That should provide you with a starting point. Obviously, you will still have to culture your sample in the appropriate media, recover the organism, and perform the appropriate biochemical tests to completely identify your isolate. So what I'm saying is that by looking at the microscopic morphology, associating where it came from, that is a clinical sample, you begin to connect the dots. Or that would help you connect the dots. Is that making sense? So these are just some examples, and does not include all. I suggest that you complete the rest of this chart. So for example, your gram stain shows you a gram-negative bacilli, starting from the left of the chart. You find that the organism looks very small, pleomorphic cocobacilli that sometimes you may think, are these cocci? But then you may find some rudsy looking organisms that some people, because of their inexperience in looking at the gram stain smear, may think that there are two different kinds of organisms present on the sample, on the smear, when in fact that is what we call pleomorphic, pleo meaning different sizes and shapes. So immediately think of Haemophilus, small pleomorphic cocobacilli, very tiny, different sizes and shape. Think about Haemophilus. Now, when you find minute, very small, very pale staining cocobacilli, and they are very much uniform in sizes and shape, and again, these are organisms or species that you could easily mistake as cocci because they're tiny and it's very difficult to see them very well. Then think of your Brucella and Bordetella in particular. There are some organisms that actually give you a very unique arrangement or shape, the curved gram-negative rods, and some of them we will describe as seagull wings, and these are the Vibrio, the Campylobacter, and the Helicobacter. So with a haystack, for example, look at the different descriptions microscopically of each of those members of the haystack group, and you will find and try to match them because they do have unique characteristics. Some of them have been described as teardrops. So look at those descriptions, and because they are not isolated or encountered commonly in the clinical laboratory, so it is more for testing purposes and being aware that certain organisms, even gram-negative rods, will show you some very unique microscopic morphology or characteristics. Now, if the gram-negative rod is a plain boring straight rod, then that is when we have to do something a little bit more drastic or different. Okay? Okay, so the routine primary culture media or setup that we call in the lab includes sheep blood agar or SBA. Sometimes we call it blood agar plate or BAP, McConkey, and chocolate agar plates for most clinical samples. However, as you have began to learn that there are certain organisms that require special growth requirements. So those special culture media will have to be added to the setup. Now, table 6-4 on page 120 of your textbook shows a selection of culture media used for different types of clinical samples. Just to give you an idea of what I'm talking about, I suggest that you visit the table, and no, you're not going to be tested, but it will help you better understand how each of the media support different types of organisms, and that will help you in learning, in your memorization of this massive content of material. Okay, so let's look at the flow chart. You may notice that the first media that I would like you to look at is growth on McConkey. Why? Because if the organism is growing on McConkey, you know that it is not a gram-positive organism because McConkey is selective. It selects only for gram-negative rods, right? But it's also inhibitory because it inhibits gram-positive organisms. So no growth on McConkey tells you that the gram-negative isolate is a fastidious organism that is inhibited by McConkey. Okay, so the negative McConkey will be for another day, but you can take a look, a quick look, that where there is no growth on McConkey, the next media that you should check would be the growth on blood agar plate, because it will tell you, give you an idea how fastidious your gram-negative rod is. No growth on blood, you then check for chocolate. So if it's growing on chocolate, probably my first suspicion, especially depending on your culture, your clinical sample, one of those organisms will be Haemophilus, because Haemophilus would only grow on chocolate. Okay. Now, if there's no growth on blood, no growth on chocolate, then you're in trouble. So, okay, let's move on the growth on McConkey. When the organism produces growth on McConkey, do the oxidase test first, and make sure that you perform your oxidase test on the colonies growing on the blood agar plate, not on the McConkey colonies. Do you know why? You have learned that the positive oxidase test produces what color? Pink to purple to dark purple, almost black. What color is McConkey? It is pink, and the colonies will produce pink colonies, and you know what I'm saying here? You'll have a false reading of your oxidase test. So the other function of McConkey agar is differential. Why is it a differential agar? Because it contains lactose. It differentiates those that ferment lactose from those that do not ferment lactose or do not ferment carbohydrates at all. So let's follow this way of thinking. As I mentioned in the previous slide, McConkey will show whether the organism is a lactose fermenter or non-lactose fermenter. So you will have two groups of organisms here based on the oxidase reaction. There will be oxidase positive that are fermenters and oxidase positive that are non-fermenters. And on the other side, you will have fermenters that are oxidase negative and oxidase negative non-fermenters. So here are some examples, and make sure that you do recognize and know exactly what you're looking at. So here's some examples of what colonies may look like when growing on Macanche. The non-fermenter bottom left also shows a greenish pigment, which is characteristic of Pseudomonas aeruginosa. Pseudomonas aeruginosa is of course a non-fermenter at all. So it is a non-lactose fermenter. In addition, it does not ferment any other carbohydrate because it has a very different metabolic process that it takes. So on the upper left, you have a non-fermenter, and the right would be a fermenter. And the middle image is a lactose fermenter with dry colonies. This is very typical of Escherichia coli or E. coli. Some of your Citrobacter and other Enterobacter species, but typically this is what E. coli would look like. But any lactose fermenting Enterobacteriaceae, they will present this kind of colonies on Macanche. On the other right is a lactose fermenter that shows very mucoid, very wet looking colonies. When you pick this up with a loop, it is very, very stringy that you'll have problems taking this colony off the culture plate. This is very typical of Klebsiella pneumoniae and is very mucoid because of the capsule that is present with this particular species. But it is not limited to Klebsiella pneumoniae, but Klebsiella pneumoniae typically will present this kind of colony morphology. Okay, so we know that Macanche contains the carbohydrate lactose, plus a pH indicator that detects fermentation. So when there's fermentation, the colonies look pink, red, dark pink. And when there's no lactose fermentation, the colonies are clear, colorless, right? It also contains bile salts as inhibitor agents. We also know that lactose is a disaccharide carbohydrate. It is composed of two sugars, glucose and galactose, make up lactose. The organism must have the enzymes required to break down lactose to glucose and galactose. These enzymes are beta-galactoside permease and beta-galactosidase. When lactose is broken down, then glucose is then available for metabolism. Remember that glucose is the simplest carbohydrate, and that is what all humans, living organisms, utilize for energy. So hence, when lactose is fermented, we can assume that the organism can ferment and metabolize glucose. Okay, some bacterial species can only metabolize carbohydrates in its simplest form, and that is glucose. I already said that, right? They cannot metabolize or ferment lactose. So the question is, when the colonies on Macanche are clear and colorless, that indicates non-lactose fermentation. But is it because that they cannot ferment lactose, or is it because they cannot ferment carbohydrates at all, or what we call asaccharidic? Asaccharidic organisms, they use or find sources, other sources for energy. So that's what we need to find out later on. When it is clear colorless on Macanche, is it a non-lactose fermenter or is it a non-fermenter? Follow? So gram-negative bacillus, that is oxidase negative, ferments glucose and reduce nitrates to nitrites, are key characteristics of the family Enterobacteriaceae. So let's follow and talk about and highlight this topic for today, this part 4 on the family Enterobacteriaceae. Wow! So the next step is this big flow chart at the end of your Enterobacteriaceae lecture, right? Okay, no worries, we'll break this down step by step. So you already have your lecture on different biochemical reactions, the kinds of results, the principles for those tests, for each of those tests. So I suggest that you go back to Micro 1 lecture notes if you are in Micro 2, and for those of you who are in Micro 1, really pay attention to those, to Dr. Farmanis' lecture on each of those biochemical test results and what is it testing, and what are the positive, negative results mean. So, but what I'm going to show you is how to proceed, so you are not just memorizing, but totally understanding, and some key things that you can use in remembering this massive maze of information. So let's begin with TSI, which is triple sugar iron agar. Triple sugar, which means there are 3 sugars in TSI. You already know that, right? Glucose, lactose, and sucrose. Review the concentration of each of the sugar present in this medium, and review why the different concentrations and what the purpose of those concentration and what that means. Yeah, I'm not gonna be repeating Dr. Farmanis' lecture. You may also find this information in the chapter in your textbook. As you can see, there are 4 possible reactions in this flowchart, excluding the K over K on the far right side. So first is your K over alkaline over acid, or K over A, plus H2S positive. Then you have an acid over acid, H2S positive, alkaline over acid, H2S negative, and acid over acid, negative H2S. But first, let's be clear what these reactions mean. So to do that, let's go back to the McConkey reaction. If the organism is a lactose fermenter on McConkey, then the TSI will be acid over acid, whether it be H2S positive or H2S negative. And if McConkey shows negative lactose fermentation, then you should expect an alkaline over acid reaction on TSI, whether it's be H2S positive or negative. So let's begin with the acid over acid H2S negative group of organisms, since this is a group that you will most likely encounter in the clinical laboratory. You look at your McConkey, and the colonies are dark pink colonies. Okay, dark pink, light pink. So you look at your TSI, and you should expect an acid over acid. So let's start with acid over acid H2S negative. So this is where you'll start on your chart. And there's a group of organisms, E. coli, Klebsiella, Enterobacter serratia. So we learned that the first test that may separate this group is what we call the INVIC, which stands for indole, metal red, Vogt-Proskauer, and citrate. And the INVIC reaction for E. coli would be positive for indole, positive for metal red, negative for Vogt-Proskauer for BP, or negative citrate, while it's completely opposite for the KES. So you can remember Klebsiella, Enterobacter serratia is KES group. And the way you should remember this is just remember one set of reactions. You know that E. coli would be indole positive. So for the INVIC, you start with positive indole, and negative citrate. Positive indole, negative citrate. And that's because we normally don't test methyl red and VP traditionally in the clinical laboratory in the traditional conventional media. Your automated and some other testing systems may test for it, but for me, the easiest way is that E. coli would be indole positive and negative citrate. And that would be the only thing that you need to remember, because you remember that KES is the opposite. KES will have negative indole and positive citrate for that group of organisms. So if you have a negative indole and positive citrate, you have to deal with KES, Klebsiella enterobacter serratia. So now the next thing that you want to look for to test is to test for motility. Klebsiella will be non-motile, while Enterobacter and Serratia will be motile at 37 degrees centigrade. And to separate Enterobacter from Serratia, you would be testing for DNase. Serratia would be positive for DNase, and Enterobacter would be negative for DNase. Now, in the laboratory, we use two media that will test three different biochemical characteristics of the organism, and that is called SIM, S-I-M. That stands for sulfide, H2S, right? Indole and motility. So with this one tube, you will be able to see if the organism is indole and indole positive or negative, and the motility test. So for Klebsiella, you should expect that it is indole and motility negative. So the SIM for Klebsiella would be negative, negative, negative. While for Enterobacter, you will know that motility would be positive for Enterobacter. So Enterobacter will have a SIM of negative, negative, positive. Okay. How about E. coli? If you look for the reaction of E. coli in the SIM, you know that E. coli is positive for indole, and it is also motile. So your E. coli would be negative for sulfide, indole positive, and motility positive. Okay? So I'm hoping that that is helping you in at least separating and grouping your organisms. So if I give you a TSI reaction that is acid over acid, H2S negative, right? And the SIM that is negative H2S, S, positive indole, and positive motility, all you need to look for is your citrate, which will confirm if it is negative citrate, then you have an E. coli. Well, if I give you an acid over acid H2S negative with a SIM that is negative, 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 that means it is negative indole and negative motility, that is and positive citrate, that will tell you that it is a Klebsiella. The same kinds of reaction, the only thing that may change, so the SIM would be positive motility, that would be an Enterobacter. If I add a DNase, that means that we're trying to differentiate Serratia from Enterobacter. Let's look at the flow chart and notice the test PAD and the list of organisms that are PAD positive. Take note of that. But before I continue with the flow chart, I will divert for a little bit. Do you know what PAD stands for? Remember that it stands for phenylalanine deaminase test. Phenylalanine is an amino acid. The test, as you have already learned, detects the ability of an organism to deaminate phenylalanine to pyruvic acid if the organism produces the enzyme phenylalanine deaminase. Now remember from biochemistry, amino acids are made up of two groups, the carboxyl group, COOH, with a minus sign, and the amine group, NH2. So certain organisms produce decarboxylases, while others do not. The carboxylase will attack the carboxyl group in the amino acid, while others will not touch the carboxyl group or the amine group, while others may not produce the carboxylases but may produce deaminases. So some organisms will attack the carboxyl group but not the amine group. Some organisms will attack neither carboxyl group or the amine group, while others will not attack or will not decarboxylate, but they will deaminate. Following so far? Okay, so let's go back to the chart. So detecting deaminase production is key in differentiating the group A group of Enterobacteriaceae. So looking at the list, when you get a positive test, as you have seen before, as you see in this chart, when the PAD is positive, you can essentially forget all the other enterics, but focus on the 3 species. PAD positive will give you Proteus species, Providentia species, and Morganella morgani. So you can remember PAD positive, ppm. Just need to remember what those ppm are. So PAD positive, Proteus, Providentia, Morganella. And just focus on differentiating those 3 groups and forget all the other Enterobacteriaceae. So let's go back to the top of the flowchart with alkaline over acid, H2S negative, and that means the organism does not ferment lactose, but ferments glucose. They are clear, colorless colonies on McConkey, and they do not produce hydrogen sulfide. So you'll see that you have a long list of species to choose from. But look at PAD. It's the first test that you can essentially eliminate or include what your organism could possibly be. So you have PAD positive, as I mentioned before, Proteus, Morganella, and Providentia. If PAD is negative, then look at the motility. Motility negative will direct you towards 2 very special or pathogenic species, the Shigella species and Yersinia. If the motility test is positive, you already know that E. coli and Serratia and how to differentiate those 2 species. These E. coli, there are certain strains of E. coli that may be non-lactose fermenter, but you don't see that as often as the lactose fermenters. But a small percentage of E. coli may be lactose negative, but glucose positive, of course. Now, Serratia is also known for certain species. Serratia are slow lactose fermenters. So in 18 to 24 hours, 18 hours sometimes, you may find your TSI to be alkaline over acid, and you may think it may be a Shigella or a Yersinia, or especially if it's PAD negative. But Serratia, if you incubate it a little longer, will eventually ferment lactose, and your TSI will appear as acid over acid. So that is a unique characteristic of Serratia. But you can easily test for Serratia with what test? With the DNase test. Okay? So let's look at the next type of reaction on TSI, and that is acid over acid. H2S positive. That means the organism produces H2S. So with this kind of reaction, you have Proteus vulgaris, and you can identify Proteus vulgaris from the rest of this list of organisms with PAD because it is PAD positive. While if it is PAD negative, you may possibly isolated Citrobacter fruindii and less commonly Salmonella arizona. One thing to remember, I believe I should mention here with Proteus vulgaris in particular, that is, Proteus vulgaris will produce clear colonies on McConkey. That means it does not ferment lactose. But when you put it on TSI, it produces acid over acid. What does that tell you? When it is clear on McConkey, which means lactose negative, but it produces acid over acid on TSI, it does not ferment lactose, but what else is in the TSI? What is the other carbohydrate? That is sucrose. Yes, Proteus vulgaris ferments sucrose, and that's what causing the TSI to appear acid over acid. On the other hand, Citrobacter fruindii looks just like E. coli on McConkey because it ferments lactose, right? So it produces acid over acid on TSI. The difference between Citrobacter and E. coli is that Citrobacter produces H2S. Make sense? Okay, so let's finish this with the last type of reaction, that is alkaline over acid, H2S positive. These are organisms that appeared clear colorless on McConkey, and your TSI is alkaline over acid, which means that these organisms do not ferment lactose or sucrose, they only ferment glucose. Okay, so here you have a list. So that's that. So now you have a list of glucose fermenters only. You have your Proteus mirabilis, Salmonella, Citrobacter, and Edwardsiella tarda. Again, look at your PAD, and once you see a PAD positive, you have eliminated or identified Proteus mirabilis. If you have a PAD negative, then you have the group Salmonella, Citrobacter, and Edwardsiella. Now, again, you see Citrobacter on two different types of TSI reactions. There are certain strains of Citrobacter fruindii that will not ferment lactose or sucrose that will appear as alkaline over acid, while most others will ferment lactose that will give you an acid over acid. So it is one of those organisms that you will see different types of reactions. But here are some key identifying something to remember with regards to differentiating this species from the others. So with Salmonella, Citrobacter Edwardsiella, here's another test in this flowchart that is called lysine decarboxylase. Remember a few slides before when I talk about amino acids, right, like phenylalanine, that some organisms decarboxylase or not. Some organisms does not decarboxylase, but they deaminate, and these are the PAD positive. And then there's some organisms, like I mentioned, produce the decarboxylase enzyme, and they're able to, I already said that, right? So here with the lysine decarboxylase test, you'll see that positive decarboxylase will differentiate your Salmonella from the Citrobacter. And then you can speciate or differentiate Salmonella from Edwardsiella with the indole test. If you wanna learn more about lysine decarboxylase, such as the use of lysine iron agar, I would refer you to figure 9-6, page 187 of your textbook. And lysine iron agar is a common test that is included in the battery with TSI, citrate, urea, sometimes PAD. So it is important sometimes to know the different kinds of reactions that you will find in the most commonly used biochemical test media in the laboratory. So I'm hoping that with this breaking down this flow chart will help you sort of sort this maze and massive number of organisms in this particular family, and the number of biochemical tests that you have to remember the reactions. So again, this is not a duplicate of your lecture material, but this is a tool to help you in learning and understanding what is going on and how to connect the dots and how to pull things together. See you next time.